Of course. So the rationale for this project was actually based on a clinical finding that we made in patients with acute gastrointestinal GVHD. So hBD-2 is an inducible defensin, which is known to be normally upregulated in response to pro-inflammatory stimuli and inflammation. And in line with this, it has already been described that hBD-2 is upregulated in the colon of patients with ulcerative colitis and also other inflammatory diseases. However, what we found then in patients with acute GVHD was that hBD-2 expression in the colon was not induced and was as low as in healthy controls. And we observed the same or even a downregulation of the beta defensin expression in mice with acute GVHD. And we therefore asked whether replacement of this defective beta defensin production using recombinant hBD-2 after allogeneic transplant could actually ameliorate disease or have an influence on acute GVHD severity. Yeah, so in the marine models, we actually found effects of hBD-2 on two major cell populations that are important players in the pathogenesis of acute GVHD. And those were neutrophils and allogenic T cells. So firstly, we found that the recruitment of neutrophils to the ileum of the mice early after transplantation was reduced by hBD-2 treatment. And we found that this effect was microbiome dependent. And secondly, we could show that hBD-2 reduced the infiltration of the intestine with allogenic T cells, which are the major drivers of acute GVHD. And using different in vitro T cell stimulation models, we then found that hBD-2 actually had direct effects on T cells and reduced T cell receptor signaling, which resulted in reduced T cell activation, and also cytokine production and T cell metabolism were reduced. Yeah, so I think that is remarkable that the prophylactic oral administration of this defensin alone had such a strong effect on GVHD severity and survival of the mice undergoing allo-HCT. And the survival benefit was even stronger than with cyclosporin A prophylaxis. And we think that this is due to the fact that hBD-2, like I said before, modulates acute GVHD at multiple different levels, including the intestinal microbiome, the neutrophils, and T cell activation. So in this study, we have only tested hBD-2 in preclinical models. However, the company that we are working with is currently working on establishing the GMP production of hBD-2 so that it can then be tested in clinical trials in the future in patients that undergo allo-HCT to then hopefully be able to reduce GVHD occurrence and severity in those patients. And our study suggests that an early intervention, so an early prophylactic treatment after allo-HCT is required to compensate for the loss of endogenous hBD-2. And therefore, we think that our data provide a rationale for the further clinical development of hBD-2 based prophylactic strategies, in particular for patients with acute GVHD. So yeah, like I said, from the clinical point of view, the next step would be the large-scale production of hBD-2 under GMP conditions, and then the testing of hBD-2 in first-in-human clinical trials. Scientifically, we are now interested in further characterizing the mechanisms how hBD-2 is actually reducing T cell activation and the T cell receptor signaling. So we have not found the receptor or the potential intracellular interaction partners of hBD-2 in T cells yet. So this is something that we would really like to investigate in more detail in the future. And we are also interested in studying whether this effect is specific for hBD-2 or if also other defensins can suppress T cell responses. So this is something that we also want to study in the future.